Hello, my name is Amy Cheung, and I'm a Senior Director of Integrated Drug Development at Certara. I'll be presenting Module 4, Case Study 3, Pediatric Drug Development, Utilization of Top-Down and Bottom-Up Approaches. Here are the learning objectives of the presentation. We will outline the challenges of pediatric drug development, highlights the usefulness of MIDD interconnectedness in pediatric drug development and how it can accelerate the process. Demonstrate the importance of knowledge building through model interconnectedness of our compound to support pediatric drug development. Finally, we will look at an example selected from the literature of rivaroxaban, a factor Xa inhibitor, to demonstrate the application of both top-down and bottom-up approaches. In general, for any recommended dose of a therapeutic agent, the sources of variability in PK and PD need to be understood. There are several specific populations for which PK can be very different. For example, the very young and the very old, and those with liver and kidney impairment. This patient might require adjustment to dose and schedule from the general population. PK study can be used to investigate this special population. In this case, PK and sometimes PD are used as surrogate for efficacy and safety, and a dose is identified that achieves a certain drug exposure or PD effect. Dose selection is key and especially challenging in pediatric development. This is because we will need to consider the differences in opportunities for PK or PD sampling, differences in the size of the body and organs, differences in the status of maturation, differences in the relevance of clinical efficacy and safety endpoints, differences in disease progression due to age. Data collected from pediatric population can also be sparse in terms of sampling point and sample size, in particular very limited for rare disease, and therefore innovative quantitative approaches can be useful to advance development in this vulnerable population with limited data. Due to all these considerations, we need to study children in much more detail and bear in mind that children are not small adults. The development over the first few years of life means that the expression of the drug metabolizing enzymes, transporter, and organ composition can be very different from that of the adult. And therefore, PK can be age-dependent in very young children. For example, the plot on the top shows how the expression of drug metabolizing enzymes changes relative to that in adults. And below, that is how the composition of the body in terms of water and fat alter with age. Many different types of modeling approaches can be applied to pediatric drug development. Here we illustrate the difference between empirical top-down modeling to mechanistic bottom-up modeling approach in understanding of pharmacokinetics. Let's focus on the schematic on the left. The most commonly used PK models are one compartment and two compartments model. All drugs are initially distributed into the central compartment. If a drug rapidly equilibrates with the tissue's compartment, then we have a one volume of distribution term. However, if the drug is shown to have a slower equilibrium to the peripheral tissue, then we'll use a two-compartmental model, and therefore we will have two volume of distribution. This kind of model is very suitable to use for population analysis to combine intensive and sparse data. This type of model is also very good in terms of understanding variability between patients and the impact of covariates. On the other hand, the physiological-based PK model will require model's details, which incorporate blood flow and tissue compositions of the organs to define PK. For quantitative system pharmacology QSP model, it represents complex system of the human physiology by integrating comprehensive biological information, and therefore it can address various purposes including target and/or disease-related biomarker identification. For the first time in human prediction, the sponsor tends to use the PBPK model to set the starting dose in phase 1. For dose projection for pediatric, population compartmental approach are mostly sufficient for age greater than 2 years old, when most of the maturation of the process, such as enzymes activities, is at the same level as adult population. PBPK will tend to be used to model data to incorporate ontogeny and differences in organ size for very young population, for example in neonates. The incorporation of QSP model has also emerged over the years, and the combination of all this top-down to bottom-up approach is getting very common to support our pediatric development program. In summary, the selection of the types of model will be depending on the objective or key questions, amount and quantity of data in terms of sample size and also sample volume, understanding of the drug PK and the robustness of the adult model, the assumption and validations plan, and the purpose of the modeling exercise, for example, whether to describe the data or to extrapolate to our unstudied population, or simulating to a new scenario. PBPK modeling enables knowledge of age-dependent changes of PK processes to predict pediatric PK. There are several commercially available platforms to enable this type of modeling. For example, SimSip has a specific pediatric modules as outlined here. There are many guidance developed specifically to focus on the choice, development, and the qualification of the PBPK models, which will help the reviewer to focus on the key requirements and areas on the choice and robustness that require for the PBPK model and the tools that are available. As mentioned, it is very important to aware the application of model interconnectedness can be used at different phases of drug development, with different types of species or population data, and various types of mathematical models. The goal is while doing this, we can optimize the knowledge we got and propagate the information to answer the key questions to advance our understanding of the drug candidate to become a safe and efficacious agent. Here we can start with quantitative system biology or pharmacology model to identify pathway activities and target engagement with non-clinical data. This will help to identify the cell lines and targets to be used in non-clinical species experiments. PBPK model is then useful to model non-clinical data and subsequently scale to human for the first time in man prediction. In the early clinical phases, POPPK and PKPD can help to understand exposure-response relationship, interpatient variability, while PBPK modeling can help to understand drug-drug interactions questions using the same human data. Both PBPK model and POPPK model can be combined to answer design questions such as sample size and also extrapolation from the reference population to target population as well. And we will now illustrate with an example. We are using an example of rivaroxaban, which is a factor Xa anticoagulant that FDA approved for treatment and prevention of venous thromboembolism (VTE) in pediatric patients and over flexible weight-based dosing. We'll show some background and highlight how model interconnectedness helped to support the dose finding in children. In recent years, there is an increasing medical need for treatment of VTE in pediatric population, especially children with severe underlying conditions, for example, children that are hospitalized who might have the risk of VTE. The guideline for the doses of VTE prevention and treatment, such as using heparin and vitamin K, are quite often due to the limited number of clinical trials in pediatric population. And these guidelines are based mainly on extrapolation from adult clinical trial results. And therefore, in clinical practice, there is a high level of off-label use of drug in children. and around 50% of the drugs that are administered to children have not been tested for appropriate age group. Rivaroxaban has been approved for adult patients for severe VTE disorder based on data from a phase 3 trial, consisting around 60 patients. As there were no existing children data of rivaroxaban back then, and with its well-defined PK/PD characteristic and efficacy safety profiles in adults, regulatory agency requests the development strategies for pediatric dosing recommendation for children less than 18 years old. The aims of the dosing strategy will aim to maintain the efficacy and safety seen in adults by using PBPK modeling to utilize existing data from all phases in adults and taking into account for the ontogeny of children to improve the study design by setting the starting dose in the pediatric study. And the aim is ultimately to reduce the off-label use of drugs in pediatric patients. This example is to show how to translate PK knowledge gained in adult development into pre-K predictions with pediatric development. And the key questions and activity will focus on data associated with a compound of interest, and modeling approach we can use is PBPK modeling and population PK modeling, with additional consideration of semi-mechanistic PD model. The ADME profile and PK/PD relationship of rivaroxaban in adults was intensively assessed through non-clinical and clinical studies, and study data was incorporated over time using population and PBPK approaches. For example, data from extensive phase 1 and phase 2 dose-ranging studies and the subsequent population modeling demonstrate that the PK of rivaroxaban are predictable and were affected by anticipated covariates such as demographic factors. The PK and PD relationship of inhibition of factor Xa activity can be described by an Emax model, and prothrombin time prolongation by a linear model. The PD parameters correlate closely with rivaroxaban plasma concentrations in both healthy subjects and in patients receiving rivaroxaban for several thromboembolic indications, including VTE prevention, after total hip or knee replacement surgery, treatment of DVT, and secondary prevention in patients with ACS. Rivaroxaban produces a pharmacodynamic effect that is closely correlated with plasma concentration that is proven by this data. This all provides important information for pediatric program and model development for rivaroxaban. The population PK of rivaroxaban has been evaluated in several population-specific models. As mentioned, it was developed as an integrated population PK model using pooled data from around 4,980 patients in 7 clinical trials across all approved indications shown in the table in the previous slide. The model was suitable to predict rivaroxaban exposure in patient subgroups of special interest. Other important understanding of the compound was also done via PopPK modeling, including being able to visualize the estimated relative bioavailability function used in integrated PopPK model by comparing it with 10 mg dose of different dose range, and the visualization of the estimated effect of the patient characteristic covariates on the apparent clearance and apparent volume of distribution was also useful. Another useful output is shown in the example here, the simulation results using the model with the area under the plasma concentration time curve from time 0 to 24 hours at steady state, following rivaroxaban 20 mg on the left-hand graphs and 15 mg once daily on the right-hand graphs for subgroups according to the renal function in the top row, age in the middle row, and the weight in the bottom row for atrial fibrillation indication. This showed how the AUC will vary in different subgroups of population, and all of this work would pave the stone again for pediatric drug development. In order to have a robust understanding of the adult data, robust model to going forward, and therefore be able to develop a robust model for POP-PK and PPPK for pediatric population. In order to understand the target exposure required for efficacy and safety, a mechanistic model was developed. By understanding the targets for anticoagulant drugs in the coagulation pathway, a blood coagulation computer model has been developed based on several published models and preclinical and clinical data of rivaroxaban. And when the preclinical and clinical data was input in the computer-generated blood coagulant model, rivaroxaban showed favorable sensitivity for clotting induced by tissue factor concentration compared with warfarin. The model also predicts a broad therapeutic windows for rivaroxaban based on a steep concentration-effect relationship that flattened rapidly at higher inhibitor concentrations. The blood coagulant modeling presented here allows robust simulation of clinical relevant blood coagulant tests and of in silico experiments that simulate blood flowing. In combination with experimental and simulated PK data, therapeutic ranges for the drugs included in the model can be evaluated and efficacy and safety limits can be determined, and hence the understanding of the PK and PD relationship. This allows dose setting in pediatric can be achieved by matching exposure between adult and the pediatric population. Now we have seen some background of the adult development information, we can recap on the key questions. How to translate PK knowledge gained in adult development into PK predictions for pediatric development. Prior to modeling, I have highlighted some important assumptions made here. As the PPPK model is incorporated with the clearing processes of rivaroxaban, one of the important physiological assumptions is that renal clearance is a function of GFR and is assumed to vary with age according to this equation. The justification is that the population is assumed to be the same as the literature used to develop the equations. It is an established literature, so is not testable. Another assumption is on disease is that the pediatric model is based on normal healthy development population, especially concerning organ development of liver and kidney. The justification is that the model may be used to inform dosing in healthy children who require major surgery and hence will be risk of VTE. This is an established assumption as parameter values from the literature. It is not testable as the PK study is in healthy children, and so the evaluation is that the model can be only used to dose in healthy children with normal development. However, once children from patient populations available, the model can then be updated to use further in those populations. The PPPK model development include the incorporations of drug and physiological informations and incorporation of maturation ontogeny into the model. And the data used are phase 1 healthy male volunteer studies PK data into the development of the adult model. This include IV studies, food effect, mass balance, trial data. The incorporation of prior assumptions and values on the PPPK model such as enzyme kinetic, transporter information, organs parameters are also needed. The diagram here outlined a workflow which summarized the general stepwise approach for PPPK model development. This includes validation from the adult model to ensure it is robust, then to extend to pediatric with the incorporation of ontogeny and maturation process. The resultant pediatric models would allow the prediction of rivaroxaban PK from adults to children in different age groups, which will impact the design and setting of phase 1 dose in the first pediatric study. The adult PBPK model is qualified using various data from phase 1 healthy volunteer as mentioned. Here are examples using IV and oral fasting data. The majority of the individual data points observed experimentally were within 90% of prediction interval. The result indicates that the adult PBPK model is well suited to describe the PK of rivaroxaban in different formulations, including tablet and suspensions, and under fasted and fed conditions, and therefore the adult PBPK model is then used to scale down to pediatric PBPK model, which will allow simulation in the pediatric population. Gender-pooled pediatric simulation for the maximum peak plasma concentration Cmax in A and B plot, an area under the plasma concentration curve AUC you can see in the C and D plot, versus body weight for high and low doses of rivaroxaban. These are simulated as oral suspension formulation compared with the adult reference formulations. Simulated data of the pediatric population are represented as geometric means, shown in blue line, and a 90% prediction interval denoted in the gray shaded area. Simulated data of the adult reference population are represented as geometric means as thick red lines, and 90% confidence interval as red shaded area in the background of the graph. The expected body weight ranges for infant, preschool children, and adolescents are indicated. The simulation for exposure throughout the investigated age range largely overlapped with the values obtained for the corresponding dose in adult reference simulation for both body weight-related dosing regimens. However, the PK values in infants and preschool children with body weight less than 40 kg were lower than the 90% confidence interval threshold of the adult reference model, and therefore indicated that the dose in these groups may need to be increased to achieve the same plasma levels as in adults. For children with body weight between 40 to 70 kg, simulated plasma PK parameters in Cmax and AUC overlap with the values obtained in the corresponding adult reference simulation, which indicated that the body weight-related exposure was similar between these children and adults. And with adolescents of weight greater than 70 kg, the simulated 90% prediction interval values of the AUC and the C 24 hours were much higher than 90% confidence intervals of the adult reference population, owing to the weight-based simulation approach, but for this patient, rivaroxaban would be administered as adult fixed dose of 10 and 20 mg. This result will help to set the recommended starting dose in pediatric trial, which is purely based on adult data and the PBPK pediatric modeled simulations. This is the result of the pediatric study based on the PBPK model predictions. Both the POP-PK and PBPK models were validated using the phase 1 study data in children and with 2 dose levels studied. The refined models are then used to predict the dose for phase 2 and 3. The diagram demonstrates the power of collating knowledge with model interconnectedness and keep refining with emerging data. The data from the phase 2 study was then provided more validation and refinement of the PBPK model and further information of the POP-PK model development. This model and simulation results will provide those justifications for label claims. In summary, the PBPK model was developed based on existing adult phase 1 data. Drug and physiological information was taken into account to predict age-dependent clearance and protein binding changes to justify pediatric dosing for rivaroxaban. Often this type of predictive model is subsequently qualified in small-scale pediatric trials where the new evidence is jointly assessed with data and pre-existing models in adults. This results in dose recommendation for children larger than often employed mg per kg based dosing. The PBPK and POPPK approach was validated with the availability of pediatric data in various phases with different healthy and pediatric patients to further impact other pediatric study design and ultimately support the labeling claims. This line shows the impact level from both sponsor and regulatory perspective and point to consider. Where interpret PK-PD chains is very important because it will help to support internal decision-making with hypothesis generations and learning through simulation and predictions. It also allowed identification of PK parameters, which is important for efficacy and safety, which will ultimately lead to dose adjustment, especially in pediatrics. MIDD and interconnectedness can drive the next decision point, but the interpretation is not ultimate, that is, the decision is likely to be revisited at a later time in the development path, for example with emerging data, and the idea is to continue validate with the new data and refine the modeling approach or combination of the approaches. In addition, using different approaches, for example POPPK, PBPK, and mechanistic PK-PD model will complement each other to support decision-making and MIDD strategy forward. And this will allow the acceleration of the program and selecting the best dose schedules for future trials. In summary, the example illustrated here shows how MIDD and model interconnectedness are essential to support pediatric R&D programs. As an aid to MIDD strategic plan development, R&D key questions with respect to the compound, mechanism, and disease or indication levels were outlined to promote effective plan creations and good practice adoptions. Important or impactful assumptions were defined, and it was shown how they were tested and evaluated. A wide range of MIDD modeling approaches through model interconnectedness were used to answer the R&D questions that produce an impact on internal company decision-making and for regulatory decisions. The utilization of various modeling approaches allowed integration of existing data from all phases, population, and various levels of quantity and sources. For pediatric investigation plan and pediatric study plan, it requires advanced evaluation and sharing MIDD plan from sponsors to regulatory authorities. And with MIDD tools, for example, assumption table and documentation standards, will help to increase the transparency and facilitate regulatory review for future pediatric plans. This allows quantitative decision-making from both sponsor and supports regulatory authority assessment, and it will ultimately accelerate medicine available to fulfill unmet medical needs in pediatric in different therapeutic areas. With this, we will have better experimental design and also omit unnecessary clinical trial or sampling drawn in children. And we will collect better quality data and use of model to understanding underlying mechanism and simulations to predict future scenarios. On behalf of Certara and Critical Path Institute, we thank you for watching this module, and we would like to acknowledge Craig, Jeff, and Mark in this. This concludes the presentation, and goodbye, and have a nice day.